So good evening everyone. So today is the fourth live session of this course RNA Biology, and as we know that in these live sessions we discuss some of the assignment problems from last year, and we basically discuss the concept behind those questions so that it's easier for us to solve similar type of other questions. So moving on to the first question for today. Coupled transcription translation is for, these are the four options that we have here: increasing rate of protein synthesis, facilitating transcription termination, a transcription initiation, and to create RNA secondary structure formation. So these are the four options for this question. So if anyone of you can answer this question, I'll wait for a few minutes. You can just type it in the chat box, or you can raise your hand and maybe answer it. And then we'll move on to discussing the question further. Anybody? Okay, so the answer for this question will be increasing rate of protein synthesis, right? So, we know about the central dogma, right? About the information flow, that DNA synthesis, DNA replicates and we get another copy of DNA. So that is the process of replication. Then we go ahead to RNA formation, which is basically transcription. Okay, and from RNA we make protein, which is nothing but translation. Protein is made up of amino acids. Okay, so that is, you know all these things already, right? So what are the players in replication? So we have the leading strand DNA, we have the lagging strand DNA, we have DNA polymerase 3, we have helicase, we have RNA primase and RNA primers, Okazaki fragments. So if you see, this is the parent DNA or the template DNA. Helicase, the role of helicase is basically to unwind the DNA double helix. Then we have an RNA primase, DNA polymerase 3. These are the SSB or the single stranded DNA binding proteins. It basically gives stability to the single stranded state. And we have the leading strand. DNA polymerase 3 is on the leading strand, and there is replication from 5' to 3' direction. Similarly, on the lagging strand also the replication is from 5' to 3' direction, right? So that is the entire process. So in case of replication DNA synthesis, DNA helicase partially unwinds the double helix at an area known as the replication fork. This is the replication fork. Okay. As the two DNA strands separate and the bases are exposed, the enzyme DNA polymerase 3 moves into position at the point where synthesis will begin. Now primase synthesizes RNA primers. Now why are they moved on the lagging strand, right? So leading strand is synthesized continuously, but lagging strand is synthesized discontinuously. Why is this? Because after DNA is synthesized, another DNA polymerase removes RNA primers and replaces them with DNA. So the gaps in sugar phosphate backbone are then sealed with ligase, resulting in two exact copies of DNA. Right? So then we see the replication. This is the structure schematic of replication. Then we have the process of transcription, which is making mRNA from DNA. So DNA basically is the stable nucleic acid that stores genetic information. RNA has three different functions, three different types of RNA. Ribosomal RNA, which makes the ribosome. Each ribosome consists of about 60%. Each ribosome consists of about 60% ribosomal RNA and 40% protein. Then you have the mRNA or the messenger RNAs that record information from DNA in the cell nucleus and carry it to the ribosomes. Then we have the transfer RNAs that delivers amino acids one by one to protein chains growing at ribosomes. In case of transcription, not all DNA is transcribed, only transcription units or genes. And each transcription unit encodes an RNA transcript or family of transcripts. So these transcripts are processed, transported into cytoplasm and translated to mRNA. Basically which is the mRNA. mRNA is made in the nucleus, but then it is transported to the cytoplasm, right? So this is the transcription unit, upstream, downstream, left to right, 3' to 5' on copied strand. In case of transcription, it's carried out by an enzyme complex known as RNA polymerase. It faithfully copies one of two DNA strands. RNA polymerase binds to a special region of the DNA sequence called the promoter, and the RNA polymerase unwinds about 1 turn of DNA, which is about 10 base pairs, to produce a single stranded template. The hydrogen bonds of the DNA are broken by the unwinding process. And the first two nucleotides are hydrogen bonded to DNA and are joined 5' to 3'. And this is continued to add new nucleotides to the 3' end. Now, as synthesis proceeds, the RNA polymerase moves in the 3' to 5' direction on the copied strand, and when a specific terminal sequence is reached, transcription stops. So, enzymes and the single-stranded RNA product are then released. So, the orientation of the RNA is determined by the promoter, that is, by which strand contains the sequence that is recognized by RNA polymerase. So, this can be oriented in either direction. So, DNA is always read 3' to 5'. So that the new RNA is made in the 5' to 3' direction. So you see, this is many RNA polymerase molecules may process the same transcription unit at the same time, right? So what is translation? But translation involves the transfer of biological information from the 4 nucleotide code of nucleic acids, ACGT, or adenine, cytosine, guanine, and thymine, into the 20 amino acid code of proteins. So each 3 nucleotide coding unit that specifies an amino acid is called a codon. A codon is a group of 3 consecutive nucleotides required to specify a single amino acid. And if we talk about translation, it has 3 easy steps. In initiation, an mRNA, a ribosome, and the first tRNA molecule, they come together. The tRNA containing methionine will recognize the start signal and bind to the P site, peptidal site of the ribosome complex. In one of the previous classes, we have discussed about the 3 sites of the ribosome. Then the process of elongation will start, right? So the incoming tRNA will bind the A site. The only tRNA able to attach is complementary to the codon of the A site on the mRNA. And a peptide bond forms between amino acids in the A and P sites, growing peptide now at A site. The ribosome complex moves so that the growing peptide is now in the P site, and tRNA from MET is in the exit and released. A new tRNA containing another amino acid binds the A site. Now then we have termination. A stop codon UAA, UGA, or UHA signals the end of the process, and an enzyme called the releasing factor binds the P site, freeing the protein. So the mRNA molecule is released from the ribosome complex, which then falls apart, and all mRNA messages are eventually degraded when the protein no longer needs to be made. Okay, so this is the translation. Step 1 is growing polypeptide chain. Step 2, you can see this entire process of translation in a schematic form. Now, what are the key parts to remember? Transcription DNA to RNA. DNA is read 3' to 5', while RNA is made 5' to 3', right? And for translation, it's basically making a polypeptide chain from mRNA. From mRNA. mRNA is read 5' to 3', and the complementary base pairing is A with T and C with G, which is adenine with thymine and cytosine with guanine. There are 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between G and C, which is guanine and cytosine. And in RNA, this thymine is replaced by uracil, basically. So, if you talk about gene expression, it's basically the process by which a gene gets turned on in a cell to transcript RNA and produce proteins. And it's basically this process by which the instructions in our DNA are converted into a functional product, which is a protein. And this process by which information from a gene is used in the synthesis of functional gene product. And these products are often proteins, but in non-protein coding genes such as tRNA and small nuclear RNA, snRNA, the product is a functional non-coding RNA. Product is a functional non-coding RNA. Now, if we talk about control of gene expression, we have regulatory genes and regulatory proteins. So, controlling gene expression is often occurred by controlling transcription initiation. So, the product of a regulatory gene is required to initiate or turn on or turn off the expression. So regulatory genes will ultimately give rise to regulatory proteins that bind to DNA to either block or stimulate transcription depending on how they interact with the RNA polymerase. Now there are different types of gene expression. For example, there will be constitutive expression. So in case of constitutive expression, some genes are essential and necessary for life and therefore are continuously expressed. So these genes are called housekeeping genes such as actin, GAPDH, glyceraldehyde 3-phosphate dehydrogenase. Or we have induction and repression. So the expression levels of some genes fluctuate in response to the external signals. Now gene expression in multicellular organisms one minute. As I was saying, there are two types of gene expression, constitutive expression and induction and repression. So in case of constitutive expression, some genes are essential and necessary for life and therefore are continuously expressed. So these genes are called housekeeping genes such as actin, GAPDH. The other case, induction and repression, the expression levels of some genes fluctuate in response to the external signals. Now, all our genes are present in every single cell, but only certain proteins are needed. So, expression of a gene at the wrong time in the wrong type of cell or in abnormal amounts can lead to deleterious phenotypes or death, even when the gene itself is normal. Now, some genes demonstrate higher expression level once being activated. It is called induced expression. Okay. On the other hand, some genes are repressed and their expression levels are lower. It is called repressed expression. So, what's the difference between exons and coding sequences? So, exons are those segments of sequence that are spliced together after the introns have been removed from the pre-mRNA. And the coding sequence is contained in exons, but it is possible for some exons to contain no coding sequence. So exons may contain sequence that is not translated into amino acids. These are the untranslated regions, or UTRs, and UTRs are found upstream and downstream of the protein coding sequence. Now, why do bacterial and eukaryotic cells have different mechanisms of gene regulation, or why the control of gene expression is much more complex in eukaryotes than in prokaryotes? The reason is eukaryotes have compartmentalization of cells. Like unlike in prokaryotes, eukaryotes having different cell organelles to perform different functions. Compartmentalization allows each compartment to perform specific functions without interference from other cell functions. Then eukaryotes have more extensive transcript processing, regulation from a distant cell and tissue specific gene expression, larger genome size, genes scattered among the genome. If we talk about gene expression, there are four major events that occur during the process of gene expression: transcription, RNA processing, translation, and protein processing. Now, if we talk about the gene control in prokaryotes, one way in which prokaryotes control gene expression is one group functionally related genes together so that they can be regulated together. This grouping is called an operon. Now, operon is one group of functionally related genes that work together in prokaryotic cells. An operon consists of a promoter, like binding site for RNA polymerase, an operator, that repressor binding site that overlaps the promoter, and the structural genes. Okay. And if we talk about a promoter, it's a specific short sequence on DNA at which RNA polymerase attaches and initiates transcription at the beginning of the transcription unit. When RNA polymerase binds to the promoter, transcription occurs. Okay. Now, operator is a specific short sequence of DNA sequence adjacent to the structural genes that the repressor protein can bind to and prevent the transcription of structural genes. So, this repressor protein is encoded by repressor genes to regulate gene expression. Now, the activity of RNA polymerase is also regulated by interaction with accessory proteins called activators. So the presence of the activator removes repression and transcription occurs. Okay, so this is basically one another schematic that talks about the DNA, which is the gene, then pre-mRNA, and ultimately mRNA, and which ultimately goes to the cytoplasm and protein synthesis or translation occurs. The eukaryotic cells modify RNA after transcription. So enzymes in the eukaryotic nucleus modify pre-mRNA before the genetic messages are dispatched in the cytoplasm. So at the at the 5 prime end of the pre-mRNA molecule, a modified form of guanine is added. The 5 prime cap, which functions as protects mRNA from hydrolytic enzymes, and a translation start point for ribosomes. At the 3 prime end, an enzyme adds 52 to 50 adenine nucleotides, the poly A tail, which acts to protect mRNA from degradation by exonucleases, facilitates export of mRNA from the nucleus, and it terminates transcription. RNA transcription and translation are the two main processing that link gene to protein. The information content of DNA is in the form of specific sequences of nucleotides along the DNA strands. Proteins are the links between genotype and phenotype. For example, Mendel's rules. Genes provide the instructions for making specific proteins. And the bridge between DNA and protein synthesis is RNA. Specific sequence of hundreds or thousands of nucleotides in each gene carries the information for the primary structure of a protein, the linear order of the 20 possible amino acids. Now, during RNA transcription, a DNA strand provides a template for the synthesis of a complementary RNA strand, and transcription of a gene produces a messenger RNA molecule. So, during RNA translation at ribosomes, the information contained in the order of nucleotides in mRNA is used to determine the amino acid sequence of a polypeptide. And the basic mechanics of transcription and translation are similar in eukaryotes and prokaryotes. Because bacteria lack nuclei, transcription and translation are coupled, which means ribosome attaches to the leading end of the mRNA while mRNA transcription is still in progress. Now, in contrast, in a eukaryotic cell, all transcription occurs in the nucleus and translation occurs mainly at the ribosomes in the cytoplasm. To summarize, genes program protein synthesis via genetic messenger RNA. So the molecular chain of a command in a cell is DNA transcription to RNA, translation to protein. Now, in the genetic code, nucleotide triplets specify amino acids. The triplets of nucleotide bases are the smallest units that can code for all the amino acids. So, in the triplet code, 3 consecutive bases specify an amino acid. The genetic instructions for polypeptide written in DNA as a series of words. So during transcription, one DNA strand, the template strand, provides the RNA template, and the complementary RNA molecule is synthesized according to base pairing rules, except that uracil is the complementary base to adenine. And during translation, sets of 3 nucleotide bases are decoded into a sequence of amino acids. So during translation, the codes are read in the 5' to 3' direction along the mRNA. The codon UUU code for the amino acid phenylalanine. The codon AUG not only codes for the amino acid ethionine but also indicates the start of translation. A specific codon indicates a specific corresponding amino acid, but the amino acid may be the translation of several possible codons, and the reading frame and the subsequent codons are read in groups of 3 nucleotide bases. So in summary, genetic information is encoded as a sequence of base triplets which is translated into a specific amino acid during protein synthesis. So mRNA is transcribed from the template strand of a gene. RNA polymerase separates the DNA strand at the suitable point and bonds the RNA nucleotide as the base pair along the DNA template. Like DNA polymerase, RNA polymerases can add nucleotides only to the 3' end of the growing polymer. Specific sequences of nucleotides along the DNA mark where gene transcription begins and ends. RNA polymerase attaches and initiates transcription at the promoter at the beginning of the transcription unit. The terminator ends the transcription. Bacteria have a single type of RNA polymerase that synthesizes all RNA molecules. And eukaryotes have 3 RNA polymerases, 1, 2, and 3 in their nuclei. RNA polymerase 2 is used for mRNA synthesis. So in eukaryotes, proteins called transcription factors recognize the promoter region, especially a Tata box, and bind to the promoter. After they have bound to the promoter, RNA polymerase binds to transcription factors to create a transcription initial complex. RNA polymerase then starts transcription. And as RNA polymerase moves along the DNA, it untwists double helix 10 to 20 bases at a time, and the enzyme adds nucleotides to the 3' end of the growing strand. Behind the point of RNA synthesis, the double helix reforms and the RNA molecule moves away. Transcription proceeds RNA, terminator sequence DNA. A transcription can be separated into 3 stages: initiation, elongation, termination. Promoter contains the starting point for the transcription of a gene. Promoter also includes a binding site for RNA polymerase. This RNA polymerase can recognize and bind directly to the promoter region. Now, as I said, gene expression is the process by which information from a gene is used for synthesis of gene products. These products are proteins, but in case of non-protein coding genes, the products are functional RNAs. It has several steps. During gene expression, the phenotype gives rise to the phenotype. Now, what are the levels of genetic gene expression? We start from chromatin remodeling or chromatin DNA protein complex, which is in the nucleus. From there, transcription, primary transcript, RNA processing, adding a cap to the 5' end and a poly A tail to the 3' end. That's a mature mRNA which comes out to the cytoplasm, and then degraded mRNA lifespan varies, and then the mRNA is converted to protein by the process of translation by the ribosome, after which post-translational modification like folding, transport, activation of proteins, all of these happens. So that is the central dogma of molecular biology. Okay, we have talked about replication, transcription, and all of that. It is symmetrical. What are the general features? DNA polymerase, gene-independent RNA polymerase. If there is a proof leading for replication but none for transcription. Okay, 5' to 3' direction, and all of that. General features of prokaryotes and eukaryotes. For the prokaryotes, there is coupled transcription translation, and for eukaryotes, transcription and translation are chronologically and areally separated. Okay. For the prokaryotes, there is no nucleus, no maturation, some directions, transcription 5' to 3', but transcription and translation is separated in case of eukaryotes. So what are the general features of transcription? DNA serves as a template. Enzyme RNA complex forms 3' to 5' phosphodiester bonds. The direction of the synthesis is 5' to 3' from the point of view from the new RNA molecule. But from the point of view of DNA, it is 3' to 5'. Substrate is nucleoside triphosphate. Beta and gamma phosphate groups are released during the synthesis. Bases in RNA are AUG, C, no T. In place of thymine, we have uracil. And there are 3 steps: initiation, elongation, termination. Now, promoter is a region of DNA that initiates transcription of a particular gene. So, promoters are located upstream of the genes they regulated. Now, what is a conserved sequence? Are conserved and located at the 10 and -10 and -35 regions upstream of the initiation site and are similar across all promoters. Okay. Now we know about prokaryotic transcription promoter, prokaryotic transcription RNA polymerase, And then initiation, elongation, and termination. We already talked about this. Rho factor dependent and rho factor independent. There are two processes of prokaryotic termination. Now, what we mean by RNA processing? The characteristic of tRNA and rRNA in nucleolytic cleavages like endonucleases and exonucleases, nucleotide addition by like tRNA, and nucleoside modification like methylation. We have talked about the genetic code, the additional genetic code. What are the features? There are 61 sense codons, 3 nonsense codons. The code is redundant, degenerate. That means there are more than needed. One amino acid can be coded by more than one codons. The code is unambiguous. A codon codes for single amino acid. The code is universal, highly conserved in the living world. Exceptions: mitochondrial genetic apparatus. The code is continuous. There are no punctuations, no overlapping. Now we talk about verbal base. It's basically the uncertainty at the third codon position. Usually the first two nucleotides are enough for the translation of the amino acid. In the first two positions, the pairing of the codon-anticodon is specific. In the third position, the pairing is less specific. Then we have the open reading frame, which is Oxfam monozystronic and polyzystronic mRNAs. Polyzystronic mRNA from prokaryotes, monozystronic mRNA from eukaryotes. Now, how to translate a codon? A table can be used to translate any codon sequence. Each template is read from 5' 3' reaction on the mRNA. Okay. Now we can talk about different types of mutations. It can be modification, like there is a mutated coding strand, there is a mutated temperature, and a mutated RNA. Or there is missense mutation, is basically different amino give rise to different amino acid sequence because there is a mistake over here, mutation, and that also leads to a mutated mRNA. Then we have modification, where mutated template strand and again a mutated mRNA. We have nonsense mutation, basically any reading, any triplet that gets transformed to basically mean the stop codon is nonsense mutation, and the polypeptide synthesis stops. We have modifications, insertion. Insertion happens in the template strand, and that's how the mutation takes on. We have frame shift mutations, where there is one nucleotide insertion and there's a frame shift change that becomes major difference in the amino acid sequence. We have deletion, some bases can get deleted, and all of that. This is a summary of the mutations. Now, what are the ingredients of translation? Ribosomes and tRNA. And mRNA, polyzystronic mRNA from prokaryotes and monozystronic mRNA from eukaryotes. So, synthesis of amino acid tRNA is by amino acid tRNA synthesis. Step 1 will be activation of the amino acid plus ATP will give rise to amino acid AMP plus pyrophosphate. And step 2 will be formation of amino acid tRNA. Amino acid AMP and tRNA will give rise to amino acid tRNA plus AMP. Okay, you can try to remember these structures because these are very important. Now, what are the steps of translation? Binding of initiation factors to the small subunit, assembly of 30S initiation complex, assembly of 70S initiation complex, uh, 30S initiation complex consists of mRNA 30S subunit, FMET tRNA initiation factors, and 70S 30S initiation complex plus 50S subunit, and all of that. Now, what are the steps of translation? A tRNA binding with the help of F2 elongation factor, GTP to A site, and peptidal transferase caused by ribozyme, and then translocation. So, what says the termination? Stop codon does not code for any amino acid. Release the ribosomal subunits, mRNA and polypeptide chain. What are unique features of eukaryotic translation? 5' cap is ribosome binding. Ribosomes can be free ribosomes, proteins of cytosol, nucleus, mitochondria, or bound ribosomes, regulatory proteins, proteins of endoplasmic reticulum, Golgi complex lysosomes, and cell membrane. So direction will be 5' to 3' end on mRNA, N terminus to C terminus on the polypeptide chain. So polysomes are formed. Now, one polypeptide chain is this and requires food energy, high energy bonds or peptide bonds like ATPA, PGT, PGDP for amino acid activation. Okay. So, what are the inhibitors of protein synthesis? Chloramphenicol basically it inhibits the peptidal transferase. Erythromycin inhibits translocation. Tetracycline inhibits amino acid tRNA binding. Streptomycin inhibits the TSF unit. Puromycin leads to early termination. Okay. So, antibiotics are a group of medicines that are used to treat infections caused by some prokaryotes. So, coming back to the question that we started with, coupled transcription translation is for increasing the rate of protein synthesis. Okay, that will be the correct answer. If anybody has any doubt, we can talk about it or we'll move on to the next question. I'll wait for 3-4 minutes. Okay, so we move on to the next question for today. What is the average percentage of alternatively spliced genes in humans? 75%, 100%, 65%, 35%. So I'll again wait for 4 or 5 minutes. You can try answering the question and we move on to the discussion. So the answer for this question will be 75%. So we have already talked about the gene expression and the transcription RNA splicing, the capping and the poly A tail at the 3' position. If we talk about gene regulation, so the molecular control of transcription in prokaryotes. In eukaryotes, transcription of eukaryotic genes is regulated by interactions between proteins and DNA sequences from within or near the genes and sometimes from distance. Right? So promoters are DNA sequences upstream of the transcripts. That initiate transcription, and promoter attracts RNA polymerase to the transcription start site. Now, what are enhancers and silencers? Enhancers stimulate transcription, silencers inhibit transcription, and both are orientation independent. Position independent, can work at a distance from promoter, can bind to regulated transcription factors, position upstream or downstream sequences. They are found in certain genes. The structure of a typical eukaryotic gene with its core promoter and proximal control region. These are the control elements. They are the proximal elements, GGC box and the CAAT box. Here the Tata box, which is the core promoter. Then there the chart of transcription, mRNA, exon 1, 2, and 3, and the poly A site. See the promoter, RNA polymerase will come and attach to the promoter. The pre-initiation complex is a large complex of proteins that is necessary for the transcription of protein coding genes in eukaryotes. They position RNA polymerase 2 over gene transcription start sites and denature the DNA. Okay, so these are the models for gene expression. Liver cell nucleus, brain cell nucleus, if you can see, control elements, core promoter, albumin gene. These are the regulatory transcription factors, general transcription factors, RNA polymerase transcription complex, the DNA and the RNA transcript. Transcription of albumin gene at high level, transcription of albumin gene at low level in the brain cell nucleus. Singular activators, these are regulatory proteins that bind to DNA at distant sites known as enhancers. And when DNA folds so that the enhancer is brought into proximity with the initiation complex, the activator proteins interact with the complex so that it increases the rate of transcription. Then we have co-activators. These transcriptional factors transmit signals from activator proteins to the general factors, and these transcription factors position RNA polymerase at the start of a protein coding sequence and then release the polymerase to initiate transcription. Then we have the post-translational transcriptional regulation, the control of gene expression regulated by the control of transcription initiation. but gene expression can be controlled after transcription with mechanisms such as RNA editing, alternative splicing, and mRNA stabilization and degradation, protein stabilization and degradation. So, RNA editing can be broadly defined as any site-specific alteration in RNA sequence during or after transcription, excluding changes due to processes such as RNA splicing. So, RNA editing is a process that changes the identity of an RNA base after it has been transcribed from a DNA sequence. RNA editing mechanisms: insertion or deletion during transcription, nucleotide, post-transcriptional nucleotide, conversion or substitution nucleotide editing such as editing AG, most prevalent in human, editing CU, editing UC. Now, what are the unmatched messages of RNA editing? The swapping out of a single base in a section of messenger RNA alters the genetic information transcribed from the DNA before it is translated into a protein. So, transcription or complementary strand of RNA is produced. A 3-base sequence codes for a specific amino acid. RNA editing, then translation, a ribosome reads the codon, and the growing polypeptide is the final protein. Relevant amino acid is added. And if the single base is altered, that is, a mutation occurs, then a different amino acid will be produced. Will be added, different protein will be produced, or a mutated protein will be produced. So, RNA editing creates mature mRNA that are not truly encoded by the genome. For example, apolipoprotein B exists in 2 isoforms. Isoform 1 produced by editing the mRNA to create a stop codon and translated to create apolipoprotein B48 in intestine tissue. Isoform 2 produced by unedited mRNA to create apolipoprotein B100 in liver tissue. Now, if we talk about post-transcriptional regulation or alternative splicing, introns are spliced out of pre-mRNAs to produce the mature mRNA that is translated. Now, splicing means removing introns or spliceosomes. Now, alternative splicing recognizes different splice sites, included some exons in different tissue types, and the mature mRNAs in each tissue possesses different exons, resulting in different polypeptide products from the same gene. An alternate splicing of transcripts makes it possible for a single gene to encode several polypeptides. Alternate splicing mechanism generates protein diversity. If we talk about the post-translational transcriptional regulation or alternative splicing, there are different factors that cause mutations in transcription factors. The protein factors that regulate gene expression via transcription are critical to many biological processes. So, when these factors function incorrectly, pathological and other aberrant effects occur. So, examples shown include development of the eye in humans, wing development in flies, and flower development in plants. Now, if they talk about the overview of eukaryotic transcription control, the regulation of gene expression or gene control is concerned with all possible ways in which gene activity can be controlled. So, while regulation of transcription initiation is the most common method of control, alternative splicing are commonly used as well. So, the control of transcription initiation can be achieved by activators and repressors that modulate chromatin condensation state. So, many other protein factors must function correctly so that the transcription occurs at the appropriate rate for proper biological function. If we talk about the E. coli lac operon, the control of gene expression by transcription, activation and repression has been studied extensively in bacteria. So, as an example, the E. coli lac operon, which encodes 3 genes, LAC-ZYA, involved in lactose metabolism, uses both mechanisms of control. A specific repressor protein, the LAC repressor, inhibits transcription from the LAC promoter by binding to an adjacent DNA sequence known as the LAC operator in the absence of lactose. So, general activator protein known as catabolite activator protein CAP binds to the site immediately upstream of the promoter, stimulating the transcription. However, the binding of CAP to its site requires the co-activator CAMP, cyclic adenine monophosphate, the concentration of which is low in glucose containing medium. Cellular conditions under which these two regulatory proteins bind to the lac promoter region are shown in the next slide, if you can see over here. So how does the regulation of lac operon transcription occur? So in the presence of glucose and absence of lactose, the transcription of the lac operon is repressed and the lac repressor is bound to the lac operator and cap is not bound to its control site due to low levels of CAMP. Now the addition of the inducer lactose to media and its binding to the lac repressor causes the repressor to undergo conformational change and dissociate from the lac operator sequence. However, if glucose still is present in the medium, CAP cannot bind to its site and transcription is relatively low. Finally, in the absence of glucose and presence of lactose, CAMP levels become high. CAMP binds to cap and the complex binds to the control site, strongly activating transcription by RNA polymerase. C. coli contains 7 sigma factors that are needed by RNA polymerase for recognition of promoters and transcription initiation. The lac promoter requires sigma 70, which is the most commonly used sigma factor in this bacteria. Now, sigma 70 combines with RNA polymerase prior to DNA binding, and the alternative sigma factor sigma 54 acts very differently in that it binds to its promoters in the absence of the polymerase. Sigma 54 RNA polymerase further requires activators that bind to upstream enhancer sequences for initiation. So these enhancer binding proteins contact sigma 54 RNA polymerase by looping of the DNA between them. So the mechanism of transcription activation by sigma 54 RNA polymerase is similar to transcription activation in eukaryotes. Now, what are the regulatory elements in eukaryotic genes? So, regulation of transcription of many eukaryotic genes is highly complex. Genes can be expressed differently in various tissues during different stages of development and under different environmental conditions. The complexity of expression of the PAX6 gene, which is important for development of the eye and other tissues, is illustrated here. So different versions of the PAX6 protein are expressed in different tissues and times of development in mouse embryos. So differential gene expression is achieved by DNA regulatory sequences located upstream, within, and even downstream of genes. So these regulatory sequences are bound by varying sets of transcription factors in different tissues. If you talk about the eukaryotic RNA polymerase, bacteria contain only one RNA polymerase, and this enzyme has a alpha 1, alpha 2, beta, beta prime, omega subunit composition. Eukaryotes contain 3 nuclear RNA polymerases that are larger but have core subunits homologous to those in the bacterial enzyme. The RPB1 subunit of RNA polymerase 2 has a C-terminal domain that is important for regulation of transcription. The C-terminal domain contains a 7 amino acid unit that is repeated 52 times. This sequence is unphosphorylated prior to initiation but is phosphorylated on serine and tyrosine residues after transcription initiation. Pol1 transcribes pre-rRNA, which is processed into the 28S, 18S and 5.8S species. Pol2 transcribes mRNA, miRNAs and 5 snRNAs. Pol3 transcribes tRNA, 5 srRNA and 1 snRNA. For initiation, there are special promoter sequences -10 and -35. Sigma factor binds to the promoter. The helices fit into major group, and in eukaryotes, several transcription factors assemble and bind to promoter. Additional sequences upstream, catbox, enhancer silence, etc. So cis and trans regulators, the cis factors are sequences upstream like Tata box, enhancers, etc. Trans factors are proteins that bind to the promoter made by other faraway genes. So chromatin structure also regulates transcription. Histones prevent transcription factors from binding In interphase, DNA is in 30 nanometer fiber phase wound around nucleosomes. So elongation is structure of the RNA polymerase. In termination, in eukaryote, special termination occurs. Enzyme cuts transcript and adds a long string of poly A tail. Now we are splicing. Critical sequences are in the introns 5' GU, branch point A, and 3' AG. So the details of intron removal from pre-mRNA molecules. So there is a cleavage at the 5' exon intron junction and formation of a lariat structure. This is the branch point, lariated branch point, and cleavage at the 3' exon intron junction and ligation of the two exons. A branch junction structure involving unusual 2' 5' phosphodiester bond between the G nucleotide from 5' branch junction and the branch point sequence. Now, what are the splicing checkpoints? Both the 5' splice site and the branch site are checked before any splicing reactions occur. So, the sequence must pass both identity checks. U1 is replaced by U6 at the 5' SS. Now, what is the splicing mechanism? U2 and U6 hold the pre-mRNA in a way that juxtaposes the 5' SS and the branch point, and the cleavage at the 5' SS and the addition of U5 bring the 3' and the 5' splice sites together. Now, what are splicing enhancers? So, there may also be special splicing enhancer sequences in the exons. Proteins and RNA bind to these sites and define the exons. Now, splicing errors can lead to disease, for example, spinal muscle atrophy and all of that. There are two genes, SMN1 and SMN2. Mutation in SMN2 can cause a skipped exon in mRNA. Now, what are self-splicing introns? So, group 1 introns in rRNA, they are able to catalyze their own splicing reactions. First example of an RNA enzyme. So, what about polymerase 1 and polymerase 3? Polymerase 1 transcribes ribosomal RNA. Ribosome is used to make proteins as two subunits, large and small. Large subunit is composed of RNA, 28S, 5' and 5S, and many proteins. Small subunit has 18S RNA and many proteins. In eukaryotes, 18S, 5.8S, and 28S RNA are adjacent, with sequence repeating hundreds of times, transcribed as a unit and then cleaved. Now, Pol 3, it makes tRNA and 5S RNA. Transfer RNAs are necessary to make proteins. They bring the correct amino acid. Each tRNA has a unique sequence and slightly different shape. All are plus minus clover leaf. Okay. Now, transfer RNA and anticodon at one end recognizes mRNA sequence. Amino acid attachment site at the other, always CCA added after transcription. Often have several modified bases, pseudouracil and ibothamidine. These modifications happen post-transcription. This is a clover leaf structure of each RNA and tRNA. Now, if we talk about polymerase 3, promoter for 5S and tRNA are within the gene sequence, not upstream. So this is the model for intron removal by the lysosome. So you see the U1 SNRNP binds to the 5' end of the intron. U2 SNRNP binds to branch point. U4, U6 and U5 SNRNPs bind to U1 and U2 and loop forms. U4 SNRNP is released, active spliceosome splicing, and excised intron sequence in lariat shape still complex with SNRNPs. Now, some genes have alternative splicing such that different exons are used in different tissues. Now, what do we talk about RNA editing? In some species, the final RNA molecule is much longer than the corresponding DNA sequence. Okay. So, if we compare the of the DNA sequences of cytochrome oxidase subunit 3 gene in the protozoan trypanosome Brucheiria fasciculata and Leishmania tarentuli aligned with the conserved mRNA for TB. Now the 5' consensus sequence is a branch point and 3' consensus sequence. Now the mRNA is cut at the 5' splice site. The 5' end of the intron attaches to the branch point. A cut is made at the 3' splice site. The intron is released as a lariat and the two exons are spliced together and the bond holding the lariat is broken and the linear intron is degraded. And the splicing mRNA is exported to the cytoplasm and translated. RNA splicing takes place within the spliceosome. The spliceosome assembles sequentially. The U1 attaches to the 5' splice site. U2 attaches to the branch point. A complex of U4, U5 and U6 join the spliceosome. U1 and U4 are released and base pairing between sequences in the mRNA and the snRNAs hold the spliceosome together. And the exons are joined together and the intron is released as a lariat. Group 1 and group 2 introns fold into characteristic secondary structures. With alternative splicing, pre-mRNA can be spliced in different ways to produce different mRNAs. This is the purpose of alternative splicing, right? So with both alternative splicing and multiple 3' cleavage sites produce different mRNAs from a single pre-mRNA. So the gRNA sequences are partly complementary to those of the unedited mRNA and pairs with it. So after this pairing, the mRNA undergoes cleavage and new nucleotides are added. Right? So with sequences in the gRNA serving as a template, the ends of the mRNA are then rejoined. So the cap consists of 7-methyl guanosine attached to the pre-mRNA by a unique 5' bond. Now one of the 3 phosphate groups at the 5' end of the mRNA is removed with the guanine nucleotide with its phosphate group is added. Now methyl groups are added to the position 7 of the base of the terminal guanine nucleotide and the 2' position of the sugar in the second and third nucleotides. The base of the initial nucleotide may also be methylated. Now, pre-mRNA is cleaved at a position from 11 to 30 nucleotides downstream of the consensus sequence in the 3' untranslated region, and the addition of the adenine nucleotides takes place at the 3' end of the pre-mRNA, generating the poly-A tail. So if you talk about what is the average percentage of the alternative spliced genes in humans, the question that we started with, the answer will be 75%. Okay. So if anybody has any doubt in this question, we can discuss or we'll move on to the next question for today. Okay, so we'll move to the next question for today. Hayflick number refers to A. Age of the cell, B. Stage of DNA replication, C. Number of times a cell can divide, and D. Cellular senescence. If any one of you knows the answer, you can unmute your mic and let me know. You can write in the chat box. Okay, so if we talk about the Hayflick number, it's basically the number of times a cell can divide. Okay. So, what is the theory of aging? So, it is basically an attempt to explain the phenomenon of aging as it occurs over the lifespan. Aging is viewed as a total process that begins at conception. So, in a sense, is a change in the behavior of an organism with age, leading to a decreased power of survival and adjustment. So, there are different types of the theories of aging: biologic, sociologic, psychologic, moral, or spiritual. For the biologic theories concerned with answering basic questions regarding the physiological processes that occur in all living organisms as they chronologically age. Okay, so what is the focus of the biologic theories? Basically, explanation of deleterious effects of leading to a decreasing function of the organism, gradually occurring age-related changes that are progressive over time, and intrinsic changes that can affect all member of a species because of chronologic age. Also, all organs in any one organism do not age at the same rate. So, any single organ does not necessarily age at the same rate in different individuals of the same species. So, stochastic explain aging as events that occur randomly and accumulate over time, and non-stochastic view aging as a certain predetermined time phenomenon. Now, what are the stochastic theories? Error theory, free radical theory, cross-linkage theory, wear and tear theory. So, if we talk about the error theory, it was originally proposed 1963. It basically means errors can occur in the transcription in any step of the protein synthesis of DNA. Error causes the reproduction of an enzyme or protein that is not an exact copy. As transcription occurs, errors to occur, the end product would not even resemble the original cell, thereby compromising its functional ability. Now, more recently, the theory has not been supported by research. Not all aged cells contain altered or misspecified proteins. So, nor is aging automatically or necessarily accelerated if misspecified proteins or enzymes are introduced into a cell. So, if you talk about the free radicals are byproducts of metabolism, can increase as a result of environmental pollutants. When they accumulate, they damage cell membrane, decreasing its efficiency, and the body produces antioxidants that scavenge the free radicals. In animal studies, administration of antioxidants postpones the appearance of diseases such as cardiovascular disease. And so, free radicals are also implicated in the development of plaques associated with Alzheimer's. Now, if we talk about the cross-linkage theory, some proteins in the body become cross-linked, thereby not allowing for normal metabolic activities. The waste products accumulate, and the result, tissues do not function at optimal efficiency. Now, some research supports a combination of exercise and dietary restrictions in helping to inhibit the cross-linkage process. The wear and tear theory was proposed in 1882. Cells simply wear out over time because of continued use rather like a machine, and would seem to be refuted by the fact that excess exercise in OS actually makes them more functional, not less. Now, what are the non-stochastic theories? Program theory and immunity theory. Right? So, what is the programmed or the Hayflick limit theory? Based on lab experiments on fetal fibroblastic cells and their reproductive capabilities in 1961, cells can only reproduce themselves a limited number of times. Life expectancies are seen as pre-programmed within a species-specific range. Immunity theory is immunosenescence, age-related functional diminution of the immune system, lower rate of T lymphocyte or killer cells proliferation in response to stimulus, and therefore a decrease in the body's defense against foreign pathogens. Now, if we continue, the change include a decrease in humoral immune response, often predisposing older adults to decreased resistance to a tumor cell challenge and the development of cancer, decreased ability to initiate the immune process and mobilize defenses in aggressively attaching pathogens, and increased susceptibility to autoimmune diseases. Now, what are the emerging theories of aging? neuroendocrine control theory and the metabolic theory, calorie restriction, DNA-related research. So basically it examines the integrated role of the neurologic and endocrine systems over the lifespan of an individual. So there is a decline or even cessation in many of the components of the neuroendocrine system over the lifespan. Now, research has shown the female reproductive system is controlled by the hypothalamus. So, what are the mechanisms that trigger changes? Adrenal glands, DHEA hormone, melatonin from pineal gland, a regulator of biologic rhythms and a powerful antioxidant, decline sharply from just after puberty. Now, the metabolic theory of aging proposes that all organisms have a finite amount of metabolic lifetime and that organisms with a higher metabolic rate have a shorter lifespan. So, rodent-based research has demonstrated that calorie restriction increases the lifespan and delays the onset of age-dependent diseases. Now, if we talk about DNA-related research, major developments mapping the human genome, there may be as many as 200 genes responsible for controlling aging in humans, and the discovery of telomeres. Now, sociologic theories of aging are the disengagement theory and personal environment fit theory and all of that. I will not go into that details. If we talk about cellular senescence, what is it, what causes it, and why is it important? So, response of normal cells to potentially cancer-causing events. So, if you see, if there's a graph on the y-axis, you have proliferative capacity. On the x-axis, you have the number of cell divisions. So, this is how it goes: a finite replicative lifespan mortal, and then the infinite replicative lifespan immortal. Okay, and this is the number of cell divisions. So what are the exceptions? Germline, early embryonic cells, or the stem cells, and the many tumor cells. So what happens when cells exhaust their replicative lifespan? So replicative senescence, irreversible arrest of cell proliferation, universal, resistance to apoptosis, stem cells, and altered function, universal but cell type specific, senescent phenotype. Now, what causes it? What causes senescent phenotype? Cell proliferation or replicative senescence is telomere shortening, DNA damage, oncogene expression, supermitogenic signals. So, what do inducers of the senescent phenotype have in common? So, cell proliferation or short telomeres, DNA damage, oncogenes, strong mitogens, potentially cancer causing. So, normal cells, mortal, cell senescence, transformation, apoptosis. So, cellular senescence is an important tumor suppressor mechanism induced by potentially oncogenic events. Most tumor cells are immortal, so many oncogenes are act by allowing cells to bypass the senescence response. Senescence is controlled by the two most important tumor suppressor genes, P53 and PRB, and mice with cells that do not senescence die young of cancer. Now, cancer is caused by genetic mutations and epigenetic tissue environment events. Now, cancer genetic events, activation of oncogenes, inactivation of tumor suppressor genes. So, inactivation of tumor suppressor genes encoding P53 and PRB proteins are the most common. And nuclear proteins controlled by complex pathways, upstream regulators and downstream effectors, control expression of other genes, halt cell cycle progression in response to inducers of senescence, and it's crucial for allowing normal cells to sense and respond to senescence signals. Now, the senescence phenotype entails changes in cell function. Aging is a consequence of the declining force of natural selection with age. If you talk about this graph, y-axis you have survivors and x-axis age, you can see how it goes. There's a term called antagonistic pleiotropy, which means that some traits selected to optimize fitness in young organisms can have selected deleterious effects in old organisms. Okay. So, antagonistic pleiotropy or cellular senescence is selected for tumor suppression, growth arrest, and functional changes. Unselected deleterious changes. Now, functional changes associated with cellular senescence, secretion of molecules that can be detrimental to tissues if not controlled. An example like senescent fibroblasts secrete proteases, growth factors, inflammatory cytokines, etc. Now, cells from old donors divide less often than cells from young donors. Cells from short-lived species are more sensitive to senescence inducers, particularly oxidative stress, than cells from long-lived species. Cells from donors with premature aging syndromes senesce more rapidly than cells from normal donors, and senescent cells expressing a senescence marker accumulate with age and at sites of age-related pathology. So this is young tissue, this is old tissue, aging. So if we come back to the question that we started with, Hayflick number refers to the number of times a cell can divide. Okay, so if we need to know about it in more detail, so basically, it's the number of times a normal human cell can divide before it stops dividing. So it's a key concept in understanding how cells age. So the Hayflick limit is commonly thought to be around 50 cell doublings, and it depends on the length of chromosomal telomeres which shorten with each cell division. So when a cell reaches this Hayflick limit, it breaks down by programmed cell death or apoptosis. So Leonard Hayflick, a professor of anatomy at the University of California at San Francisco, discovered the Hayflick limit, and Hayflick also developed the first normal human diploid fibroblast cell strains. Okay, so the Hayflick limit helps explain why cells age and why they eventually stop dividing. Okay, so telomere shortening limits the lifespan of primary human cells to a finite number of cell divisions, which is 50 to 70 for human fibroblast. This limit is called the discovered the Hayflick limit. Studies on overexpression of telomerase have proven that telomere shortening is the underlying mechanism limiting the lifespan of human cells. And since most primary human cells lack telomerase expression, the lifespan of human cells is limited by telomere shortening. Okay, so studies on human fibroblasts have shown that telomere shortening induces two checkpoints. The first checkpoint is activated in response to low levels of telomere dysfunction. 3 to 5 dysfunctional telomeres per cell. At this stage, the human fibroblasts enter a permanent cell cycle arrest, which is named replicative senescence. Human fibroblasts can escape the senescence checkpoint when T and RB checkpoints are abrogated. However, during this extended lifespan period, 10 to 20 cell divisions in telomeres continue to shorten and eventually reach a checkpoint which is named crisis. Crisis is characterized by high levels of telomere dysfunction, massive chromosomal instability, and cell death. So basically, Hayflick number refers to the number of times a cell can divide. Okay. If anyone has any question, we can try to answer that, or if not, we can conclude today's session. I'll wait for a few minutes. So there are no questions today. We can end this session. Okay. Thank you. If there are no questions, we can end this session. We'll meet again next Monday, same time, 7 to 9. Thank you.